Thank you and good evening. I'm happy here to talk to you today about 3D transdifferentiation of human mesenchymal stem cells into hepatocyte-like cells using bioprinted scaffolds. So one of the focuses in my lab is to develop different tissue engineering strategies, particularly for treatment of liver disease. Now, liver disease affects 25 million patients with over 25,000 deaths per year. It can significantly affect liver functions such as metabolism, bile drainage, glucose regulation. And when it comes to severe cases where patients actually need to have a liver transplant, there's a significant shortage in the liver donors that are available to these patients. Just to give some statistics, in 2011, 15,000 candidates were waiting on the transplant list, and only about 7,000 were available for transplantation. Furthermore, the median time to transplant, patients have to wait over a year to actually get a transplant. So there's definitely a need to find alternative methods, potentially through liver tissue engineering strategies, to help address this significant problem. So there's various challenges in liver tissue engineering. One of the major ones is that the structure and the function of liver is very complex. It has a very elaborate architecture, and it goes with the very wide range of metabolic functions of the liver. Another challenge is that it consists of several different cell types, and that's because it has these various functions. And really, one of the major challenges for tissue engineering is that the regenerative cues, multiple regenerative cues that is required to regenerate a whole organ is still unknown. And one of the other challenges is the cell source. So if patients that have liver disease, if we can obtain their cells, they're very minimum, especially in diseased liver. So the availability of healthy hepatocytes, which are the main functional cell type in the liver, is a challenge, as well as if we're able to isolate these cells, it's very difficult to expand them in culture as well as maintain their viability. And so one of the areas in my group is looking at alternative cell sources, mainly mesenchymal stem cells, as a potential cell source for this tissue target. And mesenchymal stem cells can be easily obtained through a bone marrow aspirate of the patient's own bone marrow. And there's been a lot of studies over the years of how mesenchymal stem cells, or MSCs, can be manipulated and differentiated into a variety of different cell targets, just through the manipulation of their environment as well as the addition of differentiation growth factors. And only recently there have been research and evidence that MSCs have the ability to transdifferentiate into these liver-like hepatocyte cells. However, most of the studies, actually all the studies, are in differentiating them in 2D cultures. So the effect of a 3D environment is still yet to be done, and this is what we've come to accomplished with this study. So one of the hypotheses in the study is to see what effects does a 3D environment have as far as the ability to control HMSC transdifferentiation to hepatocyte-like cells, and how do they compare to this 2D transdifferentiation. And one of the platforms that we use to create tissue engineering scaffolds are this 3D bioplotting platform, which has been explained before. This is an extrusion-based rapid prototyping platform where you print layer by layer, and you can control very well the size, the shape, the pore structure, and this results in scaffolds that have high interpore connectivity, which is very important for nutrient diffusion as well as getting a lot of cells in the actual pores. And so our lab has been able to optimize the printing parameters as well as material properties to be able to create defined structural scaffolds by printing polymers, both synthetic and natural polymers, composites, ceramics, hydrogels, and even metals and cells within these different composite systems. And we have both capacity to print at high and low temperatures. So for this particular study, we used a bioplotted polylactic acid scaffold, which is shown here. This is the actual architecture that we used for this initial study, which has an advancing angle of 45 degrees between the layers. So it's not just a grid structure, but it actually has some tortuosity inside the structure, and that's to capture more cells. We also infused a collagen slurry within the pores of the scaffold, followed by a freeze-drying step in order to create a collagen network within the pores, again, also to increase the cell seeding efficiency as well as instill greater bioactivity within the actual scaffold. And this shows a cross-section of the cell collagen fibrils within the network. So the method of transdifferentiation in these 3D structures is a two-step process where we first seed passage 5 human mesenchymal stem cells within the scaffold. And the first stage is a proliferation stage where we use a proliferating medium, which consists of fetal bovine serum, endothelial growth factor, and fibroblast growth factor for about 7 days. And then we switch out the media and treat it with a differentiation media consisting of hepatocyte growth factor, nicotinamide, and dexamethasone. And for this study, we also had a control media to compare the differences between an induction media and control, as well as, of course, the 2D control, which consisted of cells cultured on 2D collagen-coated well plates. So first we wanted to see that the media itself can make changes to these MSCs within the 3D scaffolds. And this is a day 14 time point. This is confocal microscopy of the live, which is green, and dead cells within the scaffold. And you can see that most of the cells are alive. And between the control media and the induction media at 14 days, we do see a significant difference in the cell density when they're treated with the induction media. There's a lot less cells that we see in there. However, the cells that are present seems to still be healthy. And to quantify the cell amount, we use DNA quantification. And we do confirm our images showing that at day 14, we do have an increase in cell proliferation for the control media, but with the induction media, we do get a significant decrease from day 1. And our hypothesis is that the induction media is actually, the decrease in cell number has to do with the transition from a proliferating state to a more differentiating state, which will be confirmed in later slides in gene expression. But before that, we wanted to see a more, the cell morphology, and we used scanning electron microscopy to visualize the cells within these constructs. So at day 1, the control and induction media group looked very similar. We have MSCs that are very well spread throughout the pores of the scaffold, as well as coating the struts. But at day 14, that's when we start to see these morphological changes. Here, these are flat cell sheets, again, with just a higher number of cells. But if you see in the induction media treated samples, you see a greater amount of cells that are not spread and a less amount, but you have this significant deposition of matrix, and this is a fibrillar matrix within the pores of the scaffold. So we know that there's definitely something happening in the synthesis. Biosynthesis is changing with the induction. Now, if we look at the gene expression, one of them being albumin, which is the most common protein secreted by mature hepatocytes, we do see if we, for this shows a 2D versus 3D, but the 2D and 3D cultures that were treated with the control media did not have any albumin or minimum albumin expression, which is expected because HMSCs really don't have the ability to produce albumin in their current undifferentiated state. So that was confirmed, but this graph shows 2D versus 3D cultures that were treated with this induction media, showing that at day 14, the 3D environment does in fact enhance the gene expression of this albumin protein. And this was normalized to GAPDH as well as the gene expression of undifferentiated MSCs. Now, if you look at more functional gene expression, and we looked at a keratin protein gene, which is CK8, which is present on the surface of hepatocytes, as well as a few detoxification genes, again, specific to hepatocytes. And here we show, again, at 14 days that the upregulation of these hepatocyte-specific markers is greater for the 3D culture environment. And so these results actually start to really make us appreciate the importance of a 3D environment, not just for differentiating cells, but the ability to transdifferentiate HMSCs into another cell lineage. We saw that at day 14, we start to see drastic differences in the cell morphology in the induction media, as well as matrix deposition, and that the 3D scaffold environment, with the use of these bioprinted scaffolds, can enhance the transdifferentiation of MSC into hepatocyte-like cells compared to the 2D cultures with greater expression of hepatic-specific markers. And future directions really is to look at how we can optimize the cell seating density and the scaffold architecture to enhance cell-cell contact and cell aggregation, and this is very important specifically for liver cells. The cell-cell contact is extremely important for maintaining the viability and function of liver cells, and then so we really wanted to increase this cell-cell contact, especially during the transdifferentiation phase, as well as longer-term studies, which include intracellular analysis of the components that are specific to liver cells through transmission electron microscopy, histological evaluation of liver-specific matrix molecules, as well as functional assays. Do these cells have the capacity to metabolize drugs? And with that, I'd like to acknowledge my grad students who were involved in this project, as well as our funding sources, and be happy to take any questions.